Okay, question 2. Tall plants are dominant T and they're dominant over short plants. Okay, right now we have enough to do a legend, so we're going to do that right away before we waste our time and read the rest of the question. A lot of people don't and end up having to read all the question, go back, do it again. Just for time efficiency, it's best if they give you enough information in the question to do a legend. Step 1, let's do the legend right away here. So they're saying tall plants are dominant to short plants. So, and they actually give you the nomenclature for the allele. They're saying it's a capital T for tall. So when I have one T, it doesn't matter what the other allele is. I know that genotypes are always in pairs. I'm going to put a line to simulate that I do know there's another allele, but as long as there's a capital T, it's going to be tall. Okay, and short plants, the only way I can get short plants is if I have two little t's. So I'm going to put them both in there. I'm going to make this legend very useful when I put both those genotypes or both those alleles in there. And I know that when I have two little t's, that's going to be a short plant. There's my legend. Step 1. Step 2, underline the parents. Use the Punnett square to determine the genotypes and the phenotypic ratio. So they want the ratio with the cross between a plant that is homodom, there's one parent, and a plant that is heterozygous. Okay, so homodom, genotype. Homo again means the same. So when it's dom, go back, look at my legend. The dominant trait here is tall, so that's going to be capital T, capital T. Capital T because it's dominant, homozygous, going to get two of them. Cross that with a heterozygous plant. Hetero means different. So that means I'm going to have one capital T, one little t. Of course, it's still going to be a tall plant, but it is going to be carrying the recessive allele. But because of dominancy, it's going to mask the expression of that recessive allele. Okay, so that's step 2. Step 3, Punnett square. When we have both genotypes of the parents, we do our step 3 Punnett square. Game of X is an O, cross it in. Doesn't distinguish between male and female, so we can just arbitrarily put one down the side and the other one there. So let's just put this one down the side. Capital T, we're isolating those genotypes into gametes. So these are the gametes. Half the sperm is going to have a capital T. The other sperm is going to have a recessive little t. Okay, for short. Put mum, in this case, across the top. Capital T, capital T. All of her eggs are going to be capital T. And now we just bring them together. Simulating fertilization. Put the genotype inside, or the phenotype inside. This is going to be a tall. Capital T, little t. This is also going to be a tall. Capital T, tall. And capital T, little t, also tall. 100% of these guys are going to be tall. Okay, or a ratio of 1 for tall. Okay, any questions? Give me an email and we can figure it out. Okay, thanks guys.